So again, a couple of announcements before we get started. The first one is, of course, that there's a midterm on Wednesday. That means that the material that we cover today, depending on where we get to, is going to be the extent of the material on the midterm. There will be no new material on Monday. I'll just go through and review the things that I think are either really important or that I did a really bad job explaining the first time round. So again, there's no guarantee that that will be present on Monday. There will be clicker questions on Monday based on today's lecture. So that's it as far as the midterm is concerned. I just found out that in, I think it's the first week, I think it's the 4th, remember correctly, of February, Mark Tashney will be giving a seminar over at Reed College. Mark Tashney is one of the real founders of molecular biology. He was one of the very first people to look at DNA binding proteins in terms of regulation and came up with what's called the genetic switch model that we'll talk about a little bit more later when we talk about regulation. But I wanted to give you guys a heads up about that seminar. It's at 7 o'clock over at Reed College in the evening, not in the morning. And I'll probably be heading over there to check that out. So any more questions, I'll post that on D2L as well, the information about that. Are there any questions about the midterm at this point? Yes, in the back. Oh, just stretching? Okay, that works, no problem. I won't repeat it for the record. Yeah.
When you say the big scantron, you mean the blue one, right?
Yeah, so when I say the big scantron, this is the question about the scantron to bring for the exam. Yes, the blue one. The reason for that is you can put in your student number, so make sure you know that student number, and that makes the scanning and alphabetizing and getting your scores a lot better and more likely to actually be right. So it helps you as well as help me. Other questions about the midterm? Best way to study for it is to go and look at old exams, and I do not look at the book when I put the midterm together. I just look at my lecture notes. Yeah? Question over there? Okay. Any other things? Yeah?
Are clicker questions reflective of how you ask questions?
So I'm just repeating it here. So the clicker questions indicative of how I ask questions that were likely to be on the exam. I usually take clicker questions from my list of questions that I make, some of which go on the exam, some of them show up in clickers. So they should be extremely indicative of those kinds of things. Yes? That's partly why I do clicker questions, to get you used to some of those questions. Okay, speaking of clicker questions, it's that time. About how many mutations are incorporated due to mistakes in replication per cell division in a normal somatic diploid human cell? Zero, 3, 6, 10, or 20, sorry. Ten, 5. Okay. How big is the diploid human genome? Okay, so some people ask, what numbers do I expect you to remember for the exam? the number of bases in the human genome, I think, is a reasonable number. Not the exact number, but the approximate number. So what would that be? So the haploid genome is a little over 3 billion base pairs. So the diploid genome would be about 6 billion base pairs. Okay? So that would be the kind of information I would expect you to know. So how frequently do you get mutations based on errors in replication? Approximately 1 in a billion, and that's from the lecture notes, not from the book, which would also be a good reason to think about lecture notes rather than what you're seeing in the book. So what does that mean? So 6. Most people think 3. I guess I didn't do a good job of explaining it, or there's some disconnect as far as that's concerned. So
Where does the 4 billion come from? Like 4.5
4.5
billion?
billion? So it's 3.4 billion is in the haploid human genome, plus or minus a little bit, because we actually don't have sequences for some of them, which we'll talk about right at the end of the class why we don't have sequences for that. But so you could say, you know, between 6 and 7 billion base pairs in terms of the diploid human genome. And why somatic cells? Why did I emphasize somatic cells here? Because that way they're going to be diploid as opposed to germline cells, which can sometimes be haploid. So those are also things that I expect that you know. So if that's confusing, these are terms that really are completely new to you. This is probably the right time to drop. So,
In your last lecture,
yes.
on the mutation rate slide, it actually said 3 mutations per division in humans. That's exactly
Per haploid genome, which if you listen to the lecture notes, I will have said that. So just looking at your lecture notes is not necessarily going to give you the answer to the clicker questions. Because if that were, then why would I bother? Yeah?
I was going to ask you before that clicker question, but
Yes.
there's also a table that you
Yes, there's a table.
1 in 10 billion.
Right, and that's where I also mentioned during the lecture that these are slightly different numbers.
That's fine. I'm just.
Yeah.
Is this old basically? Is the 1 in a billion?
So again, people are going to argue a lot about that. And I thought about that when I finally put this question together. The numbers are not as good as they should be here, and I will try and be more indicative in an actual exam. One of the differences between clicker questions and exam questions is exam questions get a hell of a lot better proofread than the clicker questions. But if a question like that comes up on the exam, raise your hand. I will do my best to not answer the question for you, but at least make it clear what the question is. I sometimes find during exams that I've put the wrong answer on there, and I don't know what the actual, so I don't write the key until the exam's actually done and turned in. And sometimes what I thought was the answer actually turns out to be the wrong answer. So yeah, raise your hand. Writing exams is one of the worst things that I have to do in this class. I don't like doing it. You guys probably don't like taking them either. But that's unfortunately the nature of the beast. Okay, so hopefully the next one will be a little clearer. Okay, so if DNA replicated dispersively, how many bands would Mielsen and Stahl have expected to see in their centrifuge after 3 generations of replication of DNA in light nucleotides? None, 1, 2, 3, or 4. Oh, sorry. None is A, none is A, B is 1, C is 2, D is 3, E is 4. 10, 5, okay, we got 104 this time. By the way, with clickers, you can change. It's only the last thing that you put in. So if you're running up to the end, you're like, I need to vote, vote at the beginning, go with your gut, and then think about it. So that's just clicker question idea. And I'm sorry about the bottom of the slides over here. I'll try and move them up next time. The problem is there's a little bit too much of this. So what's the key to this question? Dispersive replication, exactly. So where are we? Dispersive replication. Yeah, dispersively. Okay, right up the top there. Can't even remember my own questions. So what does that mean? It means that individual bits and pieces of DNA are being replicated separately. So you'll have pieces of new, pieces of old, pieces of new, pieces of old on both of the strands when you're doing your replication and on both of your new copies. And so what does that mean after your first, second, third, fourth, fifth, sixth, seventh, eighth, ninth generation? If you're looking at the density of the DNA, what's going to happen to it? It's going to get lighter and lighter and lighter and lighter. But it's not going to give you multiple, i.e. some heavies, some lights. And so that's the big difference here. And so what does that mean? It means no, there will be one. There will be one band. So that's all that, again, that's the idea of dispersive versus conservative versus semi-conservative. If you look at old exams, you'll almost always see one question that's kind of like this.
In the lecture notes, though, it just looked like the one hybrid band kept getting larger and larger.
Yeah, exactly, the one band.
That's not, but it's a hybrid band.
Yes, it's a hybrid band. That's exactly correct.
How is that a lightweight band?
Right, so it's not a light band. It's after the transfer to light nucleotides. So the transfer to light nucleotides is exactly what happens in semi-conservative replication. So transfer to light nucleotides, after one generation, how many bands do you see? In regular semi-conservative, one. After 2 generations, how many do you see? 2. After 3 generations, how many do you see? 2, exactly. And you will continue to see 2. So, but that's because you've got an old strand and a new strand, which is then half of the density. And so in dispersive replication, you're going to have half old, half new. That's also going to give you one band. Then what's going to happen at the next one, though? You'll have 1/4 old, 3/4 new. And then you'll have an 1/8 old and 7/8 new. The band is going to shift. It's going to become less and less dense, but it's still going to be just one band. So the idea here, now this may seem like a really silly question, why bother with these kinds of things? The idea is to hopefully get you to think about the process rather than know the answer, because biology changes constantly. And the idea here is that hopefully I'm helping you think rather than helping you memorize. So that's the theory. Practice, again, you can let me know all about that when you do your course evaluations at the end of the term. So that's my exam. Okay, so last time, basically, we talked about pretty much everything that's on this side here. Now we're going to talk about everything else that has to do with replication over here. And again, wherever we get to is where we get to today. By 11:20. So start out a little bit with the replication fork. Talk about the sliding clamp, also known as the processivity factor. And then the replisome, which is putting together all the components that you have at a replication fork in order to get replication to take place. It's not just the DNA polymerase by any stretch of the imagination. You need a lot more in terms of getting replication to take place. Talk a little bit about mismatch repair. That's how you get the last little bit in terms of fidelity of replication. Supercoiling, which again we may have a chance to talk about, has to do with the fact that since we have these two strands that are wrapped around each other, you only can get replication once those two strands have been pulled apart. So you pull apart the strands. That's what the DNA helicase does, one of the absolutely critical components of a replication fork. But when you pull apart two strands, what happens is, since they're wrapped around each other, you're unwrapping them. And in that unwrapping process, you end up generating lots of other wraps. And so that turns out to be a real problem in terms of making sure that you can get replication, or for that matter, transcription, to take place. And so you have to have specific enzymes to deal with that, and that's also part of what's present at the replication fork. Talk a little bit about some of the eukaryotic origins and replication, but again, we'll see how far we get there. This is exactly the same slide that we had last time, but I wanted to spend a little bit more time talking about what happens here on the lagging strand, or what also happens right where you start replication, even on the leading strand. You have to start with an RNA primer, because DNA polymerases can only extend, they can't initiate. So you have to have an RNA primer, an RNA polymerase that puts down your primer. And then that primer, A being RNA, but much more importantly, it's low fidelity, i.e. there are a lot of mistakes there. You need to make sure that this RNA is gotten rid of. The process that does that is RNase H, and then DNA ligase. And that's shown a little bit better here. This is a figure that you'll have a lot of trouble finding in your textbook because it's not there. It's from the Molecular Biology of the Gene. It's the Watson textbook, which we used a couple of years ago. I don't have it on reserve in the library yet, but it will be there. But basically, it's just another way of looking at exactly the same thing here. So RNase H, what does RNase H do? It will cleave RNA in an RNA-DNA hybrid. That's what the H stands for, is the hybrid. It will cleave this, but it always leaves a little bit, and sometimes more, sometimes less. So even you see this green thing here, that's one nucleotide of RNA, and often there'll be a little bit more than that. This needs to be removed. How does it get removed? It gets removed by 5' exonuclease. Where does that 5' exonuclease come from? Turns out that 5' exonuclease actually comes from DNA polymerase I. Well, that's bizarre, because what do DNA polymerases do? They start at 3' OHs and extend from 3' to 5', right? So that makes sense, and they've got proofreading, which is going backwards. So that's your 5' to 3'. But then why should you be degrading in the same direction that you're polymerizing from 5' to 3'? So that's very strange, but it turns out it's absolutely critical for getting rid of some of these last nucleotides here. So DNA polymerase I, as well as having a 5' to 3' exonuclease, also has a 3' to 5' exonuclease. Proofreading and for removing primers here. So once you've removed this primer, that's my pointer here, remove this primer, often you will also be doing this again with one of these DNA polymerases that will polymerize all the way to here, but now you have your 5' phosphate and 3' OH that you can't hook together here, because all the polymerase does is adds nucleotides. It doesn't hook up those that are already there. And for that, you have to have a DNA ligase. And so DNA ligase is what brings everything together here, so you finally have your continuous DNA strands. So starts out semi-discontinuous because you're making the lagging strand in individual pieces, but eventually, of course, it has to be continuous, so you've got both double strands there. So how do you make these? Okay, clearly if the RNase H is giving you a template and a primer, that's fine, but how do you start? You've got to get the two strands apart. That's the process of the DNA helicase. I like to think of this as sort of a giant donut. These DNA helicases wrap around a single strand of DNA, and sorry for the people who are sitting over here. There's a nice image of it down in the corner of the slide here. But basically, it's kind of a donut which goes around a single strand. And that single strand, as you can see up here, then basically gets pulled apart from the other double strand through lots and lots and lots of ATP hydrolysis. And in fact, in terms of replication, the ATP hydrolysis by the helicase is probably the main use of ATP in terms of the whole replication process. So this burns huge amounts of ATP, is just pulling those two strands apart, and that was why people had trouble with the whole semi-conservative replication model, was, how do you pull these two strands apart? Actually pretty stable. That's what Watson and Crick came up with in their whole structure. So the presence of the helicase is really important and uses a lot of ATP hydrolysis in order to get the two strands apart. The next problem is once you pull these two strands apart, what do those bases want to do with each other? Bind back to each other. And so you have to have a way of, once those strands have been pulled apart, with all of the energy that's been expended by the DNA helicase, you have to have a way of keeping them apart from each other. And so the way that happens is through something called the incredibly creatively named single-stranded DNA binding protein, or SSB, single-stranded DNA binding protein. So this will bind to single-stranded DNA in a non-specific fashion, because you don't want to have very specific binding here. You just want to be pulling the two strands apart. And also binds in what's called a cooperative manner. We haven't talked too much about cooperativity yet. Hopefully, Dr. Bartlett talked about this a little bit when he talked about chromatin and chromatin remodeling complexes. The basic idea is once you have binding by one of these DNA binding proteins, it stimulates the binding of a whole bunch more. And so basically the rate limiting step here in terms of your interaction is the binding of the first one of these proteins to your single-stranded DNA. And once it's bound, then through protein-protein interactions, it will bind then very, very quickly to all the rest of it. And cooperative binding is a very, very common occurrence if you think about DNA binding proteins, and for that matter, many other interactions which you have present in the cell. So as well as keeping the two strands apart, which are complementary to each other, this single-stranded binding protein is also very important for removing so-called secondary structure in the DNA. And the secondary structure in the DNA is just when the two strands come and bind to each other. So if you ever have a sequence in the DNA which is complementary to another sequence, that could also bind together. That would cause a problem for the DNA polymerase. That's not the only thing that these do. It turns out that single-stranded DNA is actually really quite unstable. And one way to make it more stable is to associate it with a protein. So the single-stranded binding protein is also very important for that process. So we've got two strands pulled apart. They're bound to single-stranded DNA binding protein. Now we need to replicate that. What's going on with this DNA polymerase? The little Pac-Man. Anybody remember Pac-Man? Anybody old enough to know about Pac-Man? So process here. So here's our DNA polymerase. Going along again, it's got a nice template. Here's our primer. Got a 3' OH. Can extend all the way along here. Unfortunately, DNA polymerases by themselves are actually pretty crummy at staying on the DNA. And what I mean by that is that they have very low processivity. And what that means is that the DNA polymerase will add a nucleotide and in many cases fall off of the template. No template, no replication. So this is a real problem if you're trying to replicate something even as small as the E. coli genome with about 5 million base pairs, or something as big as the human genome with your 6 billion, we're talking about the diploid genome. So how do cells deal with this? Well, what they do is they have what's called a processivity factor. Processivity factor is also known as sliding clamps. It's basically a big ring that goes around double-stranded DNA. So this is now different than the helicase. Helicase just goes around a single strand of DNA. The sliding clamp structure goes around both strands of DNA. Again, it makes a ring structure shown here as this sort of bluish, purplish ring. Here's, in fact, a high-resolution structure of that particular protein. This one is the one that we have in eukaryotic cells, also called pCNA. Anybody know why it's called pCNA? Horrible name. Proliferating cell nuclear antigen. So proliferating cells, what does that mean? You're replicating. Nuclear antigen, antigen is something that's interacting with antibodies in the nucleus. What that means actually is it's a really pretty common protein. You've got a lot of this protein in replicating cells. And it makes sense, certainly in the case of keeping the polymerase onto the DNA, but in theory you should only need one of those, right? Well, it turns out you also need them a lot on the lagging strand, and we'll see that a little bit more in just a second. Turns out that the structure of pCNA has this interesting alpha helical structure here, not surprisingly, with a number of positive charges on it, interacting with the DNA, and then these beta sheets around the outside. There's the equivalent protein in bacteria. It's called the beta protein. Interestingly, pCNA has 3 subunits, so it's a trimer. It's a little hard to see here. The colors should be a little bit different relative to each other. These two are one protein. These are another protein. These are another protein. This viral protein from bacteriophage T4 also has 3 subunits. But curiously enough, the bacterial protein has 2, even though the structure is almost identical here. And what that means is there was probably some kind of really basic subunit here, which then got copied multiple times through evolution. In bacteria, you had 2 of them. In eukaryotes, you ended up with 3 proteins, which were doing the job here. This sliding clamp, in order to slide along the DNA, does not require ATP, but putting it onto the DNA does. And so that process, you have to have something, again, very creatively called the clamp loader. So sliding clamp, which puts that basically donut around the DNA. It's got to open it up, put the DNA in, and close it again. It's the process shown up here. I'll get to you in just a second. Here's the process in bacteria, because your sliding clamp just has 2 subunits. The clamp loader will open up the clamp, put it onto the DNA. ATP hydrolysis happens. This clamp loader falls off. You have the DNA polymerase, which associates with the sliding clamp. And now this is a processive polymerase, and it will stay on the DNA. Yeah?
How does the sliding clamp keep the DNA polymerase from falling off?
So how does the sliding clamp keep the DNA polymerase from falling off? Great question. It's on the next slide. So the mechanism is that it's a protein-protein interaction between the DNA polymerase and the sliding clamp. And we know this quite well, turns out, for the eukaryotic pCNA. One of those 3 subunits that comes together to make pCNA is really good at binding to DNA polymerase. And so basically what happens is all of these DNA polymerases are non-processive. They'll fall off of the DNA. But the protein-protein interactions that they have with the sliding clamp are much stronger. And so basically, all that the sliding clamp does is prevents the DNA polymerase from escaping away from the DNA, and so it can restart and keep going. Yeah?
Does it ever fail and lose the polymerase?
Pardon?
Does it ever fail and drop the polymerase off the replication strand?
So does it ever fail and drop the polymerase off of the strand? So people didn't think that that was the case until actually really quite recently, and it looks as if there actually probably is a little bit of that going on. And so sometimes it'll fall off. But the protein-protein interactions between the sliding clamp and the DNA polymerase are very strong. They have very low dissociation constants. And so they do stick together extremely well. Now that's what get back to that in just a second, because there are a couple of really interesting aspects about the replicative DNA polymerase versus the non-replicative DNA polymerases. Replicative polymerases are the ones which are going to be making those strands. The non-replicative DNA polymerases, we'll talk a lot more about later, but we already talked about one of them, get back to in just a second, don't interact with this. And so those are non-processive. They fall off the DNA all the time. So if you don't interact with a sliding clamp, you're going to fall off the DNA all the time. If you do, you're going to stay on the DNA. Yeah?
How does it know when to, like, let it leave?
How does the clamp know to let it leave? Okay, right down here at the bottom. Thank you. So when the polymerase gets to a primer or some part of the DNA that it can't replicate anymore, because down here there's no template, then there's a dissociation that takes place. Yeah, there's another question over here? Another hand? Or was that sort of the same thing? Okay, so that's the process, and this is again just an image of the beta sliding clamp, giving you an idea of the size of the protein. And of course, it's right here. That's where it's associating with the replicative DNA polymerase. So let's try and put most of this together. There's still a few things that are missing here. But this is what a replication fork basically looks like. Here we have a DNA helicase pulling apart the two strands. We'll start here by the one at the top. This is your leading strand. The leading strand has a sliding clamp on it and a DNA polymerase. This is perfectly happy. It's got a template, got a primer. We're good to go. The lagging strand, of course, is a little bit more difficult, because all of the DNA polymerases have a 3' OH and extend from 3' OHs. So they're always going to be going from 5' to 3'. And of course, that opposite strand has gotten flipped around. So we already talked about semi-discontinuous, but the DNA polymerase is in fact together with the replication fork here all the time. The replicative DNA polymerase is actually very small amounts of those replicative DNA polymerases. So they're all associated here at the replication fork. So you don't have lots of DNA polymerases here on this lagging strand. You just have one. And what that means is that this template for the lagging strand has to have something associated with it. First, of course, a single-stranded binding protein. And now, replication again. Sorry for people over here. We've got our primer, which is going to be extending here, but you have a long bit of unpaired and unreplicated template, which again is bound by your single-stranded binding protein. Your DNA primase, which is going to put down a primer. Now you have to add a sliding clamp here, because you need to tell the polymerase this is the place that you've got to start. So putting on a sliding clamp, you have to do this for every single one of the Okazaki fragments. And this is why you have a lot of proliferating cell nuclear antigen, because for every one of those Okazaki fragments, you've got to be putting on one of these sliding clamp structures. And what this means is that this strand ends up getting longer and shorter, and people talk about this as the trombone model for replication. And we'll take a look at an animation of that in just a second, because it's a lot easier to see, or at least for me to visualize, when you actually see a process going on here. So we've also got these primers, again shown here as blue dots. That's the RNA which we need to get rid of. So those are some of the things we're actually missing in this particular view of the replication fork. There are now electron micrographs. You can see what the replication fork process, the whole machinery here looks like. The only message here is that it's really big. There's a whole bunch of stuff here. And you can also see this loop structure. This is an electron micrograph. Here's your double-stranded DNA. Gets separated. Here's the leading strand, and then the lagging strand with this big loop here, which is the template on which you're going to be putting all of the Okazaki fragments. It's just another way of looking at that. We flipped it around a little bit. Here's our helicase RNA primer with the primase being made right here. The sliding clamp, which has to get loaded by this clamp loader complex, which then will allow the lagging strand polymerase to extend until it gets to this primer. Once it gets to this primer, what happens? The polymerase falls off, but now it's attached here to everything else. It can bounce onto this primer right here, extend that primer, get to the end, and go through the whole process. This is yet another way of looking at that process. Again, here's our helicase, the primase. The clamp loader is really the important thing, not just for putting the clamp on, but also holding on to the leading and lagging strand polymerases, which are present at the same time. This is just showing the process. Here we have extension. That was the primer that was made there. Here's our single-stranded binding protein. Once the polymerase gets to the next primer here, it will dissociate from the sliding clamp, and then, since it's held down by the clamp loader complex, back to the next sliding clamp and extend from that one, which you can see here, and go through this process again and again and again. Yeah?
Do the same proteins copy the entire strand of DNA, or is there kind of a shelf life of a certain number of base pairs before they switch out with the new helicase just break it apart? Or...
Okay, so basically the question is it always the same complex that replicates through the genome, or is it associate and dissociate all the time? I'm not sure, we haven't really talked about it too much yet, but there are 3 main polymerases, DNA polymerases in E. coli. We already talked about DNA polymerase I. That was the one that Roger Kornberg found, but also has this funky extra exonuclease activity, proofreading, and the counterproductive process. There's also DNA polymerase II, which nobody really knows very much about, and DNA polymerase III. And it's DNA polymerase III which is what makes the whole genome. And it turns out that in the case of E. coli, where you've got 2 replication forks, each of which is going to have a leading strand polymerase and a lagging strand polymerase, you can actually get away with just 4 DNA polymerase III molecules and replicate all the way around the genome. And so you're not actually assembling and disassembling. Now, getting back to why DNA polymerase III, it's the 3rd one they found. Why was it the 3rd one they found? Because there's a lot less of it. If you just need 4 copies of it for your, replicating your whole genome, you don't need very much of that particular one. Now that, of course, is the one which associates with the sliding clamp. The DNA polymerase I does not. If you look at a eukaryotic replication fork, basically you need all the same activities, but just to make things confusing and fun and interesting, they were all given different names. Thanks. Okay, but the same thing has to do with the polymerases. So here, instead of calling polymerases 1, 2, and 3, the eukaryotic people decided we'll just use Greek letters instead. So we've got alpha, beta, gamma, delta, epsilon, and actually I think there are about 10 different DNA polymerases. The important ones for us are Pol delta, Pol epsilon as well, but in this case Pol delta. Pol delta is the replicative polymerase, so it's just like the DNA polymerase III that you have in E. coli. That's the one that associates with the sliding clamp. The sliding clamp here is now pCNA, not beta. Also have a clamp loader, and these are, again, confusingly named, but unfortunately this is what you see in the literature and might show up on exams. The replication factor C. Replication factor C is the clamp loader. Replication protein A is the single-stranded binding protein. And this, again, is basically more or less identical. There's a DNA helicase, topoisomerases that we'll get to in just a second. What's more different is what's going on in a lagging strand. So in eukaryotes, you still have a DNA primase because all the DNA polymerases are the same way. They can only extend at 3' OHs. They have to have an RNA polymerase to get a primer. So we've got this primase, which is an RNA polymerase. But then Pol alpha binds to the primer and extends the primer. So it's basically just making your primer a little bit longer before Pol delta comes in and extends the rest of that, and that's what's shown over here. Then you have primers. You need to get rid of primers. You have RNase H, just like you have in E. coli. But then you have this other strange protein, and again, sorry for people who are over here on this side, called FEN1. FEN stands for flap endonuclease. Well, endonucleases, we all know what those do. They cut in the middle of your nucleic acid. But what's the flap? Well, the flap, it turns out, is the RNA primer, which gets pushed out of the way by the DNA polymerase. And so the flap is this RNA which is flapping around. And the flap endonuclease will cut that off. One big difference here, and this is a big difference in the DNA polymerases between E. coli and most bacteria and the eukaryotic system. The DNA polymerase I in bacteria has the 5' to 3' exonuclease activity. It can chew up this last piece of primer here. Pol-alpha and all of the eukaryotic DNA polymerases don't have this activity. So they do have to have some other way of getting rid of that last little piece of RNA, which is here, and that's where the FEN1 endonuclease comes in. It's the flap endonuclease. This might be a good thing to look at when you're thinking about studying for the exam. It basically gives you comparison of the different proteins and the nomenclature for them. So DNA polymerase III in bacteria, which is the replicative polymerase, in eukaryotes, DNA polymerase delta. The primase is also primase, but in eukaryotes you have DNA polymerase alpha, which helps with the primer. DNA B is the helicase in bacteria. MCM, I actually should get rid of this question mark here, is the replicative helicase. Single-stranded binding protein is replication protein A. The beta sliding clamp is PCNA. The clamp loader is RFC. So basically need all of the same activities, but this is the different nomenclature, which you say, unfortunately, is what has happened. So again, I mentioned before, looking at the replication fork, I find it's a lot easier to look at in an animation. This is just the link to that. Here's the molecular visualization of DNA. Actually, hopefully it will run for us. Click. There we go. So this is DNA replication. Again, I've given you the link here. This is the replication complex right here, where we have everything together. There's our DNA helicase. This DNA helicase is pulling these two strands apart. Here we have our leading strand polymerase together with its sliding clamp. And now what's important is we need to look at the lagging strand. Here's the lagging strand over on this side. This is the polymerase, which will jump from here. You can see it much better in this case. We've got the single-stranded DNA here. The polymerase starts on that, will polymerase from 5' to 3' until it gets to a primer, fall off, and then reassociate with the next sliding clamp, which has been added to it. So I recommend taking multiple views of this. I usually have to take a bunch to take a look at it. But this is that extra single-stranded DNA here, which is made until this lagging strand polymerase gets to the next primer here. Okay, yes, no, huh? Totally confusing process. Again, I recommend taking a couple of looks at that. Okay, are there questions about that particular aspect of replication? Yeah?
So on the diagram you had the bacterial replication, that's where the clamp loader had the two polymerases on it. In eukaryotes, are they like connected like that too?
Okay, so the question is, for the two polymerases, the leading strand polymerase and the lagging strand polymerase, are they also connected in eukaryotes as they are connected in bacteria? Sorry, I'm just paraphrasing your question. Yes. And so at replication forks, you always have the leading and lagging strand polymerase associated with each other through protein-protein interactions with each other. Other questions? Yeah.
In one of the slides, you said that in bacteria, DNA polymerase III was the equivalent of poly delta.
Mm-hmm.
And I thought that polymerase I was responsible for replicating the leading strand in bacteria.
Okay, so the question here is basically what's involved in making the leading versus lagging strand in bacteria. Is it DNA polymerase I or is it DNA polymerase III? And so the answer to that is that both leading and lagging strands are made by the replicative polymerase, which is DNA polymerase III. DNA polymerase I is involved in dealing with primers. It's just getting rid of those last little pieces that are present in the DNA. And if you think about it, there are a whole bunch more primers that you have to get rid of than you have to have just at the replication fork to replicate your way through, which is why the Kornberg lab originally found DNA polymerase I, because there was more of it. Yeah?
So, and that was what you just said is true for eukaryotes and bacteria for the DNA polymerase I?
Okay, so anytime it's DNA polymerase I, II, or III, it's going to be bacteria.
Okay.
Anytime it's DNA polymerase Greek letter, then it's for eukaryotes.
Okay. So then, sorry, at what point, so like at the start of this lecture, was where you started talking about the replication fork with the lagging strand and initiation, was that just for bacteria or is that a generalized slide for both? I'm trying to differentiate.
So
just the bacteria slide.
the question here is basically how general is our replication forks and this sort of replosome complex? And the answer is extremely general. It's true for bacteria, true for eukaryotes, true for archaea, true for most viruses, at least the DNA viruses. So you've always got helicases pulling apart the two strands. You're always going to have a leading strand polymerase and a lagging strand polymerase, which are always the replicative polymerases. They're attached to each other. They're always sliding clamps. There are always going to be clamp loaders. So that process is the same across all cellular life that we know of.
Okay. And so you differentiate once you get, okay.
So it's basically the differentiation is in the names, but the activities, because the process, pulling apart the strands, replicating it, is the same. No, DNA is present in all cellular life. And that's always being replicated. You always have the same kinds of problems, the same kinds of issues that you need to deal with. Again, that probably means that this is something that's really, really old and has been, again, evolved for probably billions of years. Yeah?
So we saw the detailed process
Hmm?
in eukaryotes and in bacteria, but it also mentioned a sliding clamp difference T4 for the bacteriophage virus. Should we know any of the other names for any of the other parts for the viruses?
Okay, so the question is, should you know the names for any of the parts other than the T4 sliding clamp for viruses? Have I mentioned any of those in lecture so far? No. What do I base my exams on? What I mentioned in lectures. So what does that mean about knowing other proteins? No, you do not need to know them. But then you take my virology course next term, and then we can learn all about them. But no, you don't need to know them for Wednesday. Okay, so let's talk a little bit about supercoiling. We could talk about supercoiling ad nauseum probably here, at least as far as people are concerned. But the big problem here has to do with when you have DNA helicase, what happens? You unwrap the DNA, and there are two strands that are wrapped around each other. As you pull apart two strands, you're going to have problems that develop because, and unfortunately I don't have my double-stranded helices here, but if I could pull apart those two strands, what happens is you pull apart two strands that are wrapped around each other, you're going to end up generating compensatory either wraps around each other, but what happens if you've got these things that are wrapped, they'll end up writhing around each other. And so I can sort of show this a little bit here. And they've got our phone cord. Again, nobody has these anymore. But if you take this and twist it a little bit, see it's over-twisting, maybe a little hard to see at the top there, but it's over-twisting. The way to deal with that is then it wraps around itself. And that's exactly what happens with DNA when you pull the two strands apart. Ends up wrapping over itself. And so this is the process shown down here at the bottom. Our DNA helicase is pulling these two strands apart. What happens? You end up with these wraps that happen in the DNA in front of the replication fork. And what happens is, if you more and more you pull apart here, you're going to end up generating more and more of these extra windings in your DNA. And it turns out if you don't deal with those, the polymerase stops. It can't get through this extra unwound DNA. So you have to have another way of dealing with that, and that's shown here as what's called a topoisomerase, because it changes the topology of the DNA. There are two flavors of topoisomerases, topoisomerase I and topoisomerase II. The only thing you need to remember about those is that topoisomerase I cuts one strand and topoisomerase II cuts two strands. Yeah?
Does DNA polymerase move as fast as the helicase?
Does the DNA polymerase move as fast as the helicase does? So the answer to that is yes and no. So it turns out that DNA polymerases in eukaryotes are wimps. They go really slowly relative to the DNA polymerases in bacteria. DNA polymerase in bacteria does go about 1,000 nucleotides a second. DNA polymerases in eukaryotes go about 1/10 of that speed, so about 100. That may well have to do with the fact that in eukaryotic DNA, what did Dr. Bartlett talk about? Nucleosomes. All this stuff is much more packaged in eukaryotic DNA, so that's probably why they're going a lot slower in that case. So this is just showing, again, basically exactly the same issue. Here we have our DNA helicase. The DNA is being pulled apart. It's going to end up being overwound. Now, if the end of your DNA is actually free, what happens is it just spins around a lot, which is fine if your DNA is free. But it's not free because you have to compact it. It's always going to be attached to parts of your nuclear lamina. It'll be attached to the other end of your DNA. You think about bacterial chromosomes. They're actually circular chromosomes. So there's nowhere for this extra overwinding to be taken care of. So what you need, as I mentioned before, are topoisomerases. Two flavors of these, topoisomerase I and topoisomerase II. Topoisomerase I cuts one strand of DNA, and basically what it does is it's sort of the reverse of that unwinding. You've been unwinding it. It needs to wind itself back up. If you cut one strand, then that one strand can rotate around the first one and end up back where it started. And in that process, you've basically undone the unwinding process. Topoisomerase I's are really fascinating enzymes, but mostly because what they do is they have a tyrosine in them. Tyrosines are one of those interesting amino acids that you might want to remember as far as Wednesday is concerned, because they have OHs sticking out at the end of their side chain. Why are OHs so important? What do you have in DNA? You have OHs. And so what the topoisomerase I does is basically tricks the DNA, one way of thinking about it, but this OH then gets hooked up to a 5' phosphate. If it's now hooked up to a 5' phosphate, you have hydrolysis of this bond in the DNA. Now, these two strands, one of them is no longer attached. So it can rotate around and then be religated on the opposite side here. And so this interaction between the tyrosine OH on the topoisomerase and the 3' OH that you have in the DNA, these are almost isoenergetic. They're very, very similar in terms of their binding and activity. So you don't need any extra ATP hydrolysis to get these reactions to take place. So it's just an exchange of your OH on the tyrosine with the OH on the DNA. Yeah?
You said primase comes in and refills that?
No, so this is just a reversible reaction. So it's just the topoisomerase. Just the topoisomerase itself. You don't need anything else there. And so the topoisomerase is basically acting as an endonuclease and a ligase. So when it binds to this strand, it will cut that phosphodiester backbone at the one position, but also this reaction is easily reversible, and so it acts as a ligase and puts it back together. Yeah?
So is the binding actually like a condensation type reaction where a water molecule, how does it, two OHs come together? Is that what I'm...
No. So OH interacts with phosphate. So it's the OH on the tyrosine will bind to the 5' phosphate, and then that will reverse. The 5' phosphate will then hook back up to the OH, which is there. Okay, so this is topoisomerase I. It's just breaking one of the strands, allowing it to wrap around and come back together. But we also have double-stranded DNA, and as I mentioned here, when you overwrap something, it ends up giving you multiple strands that are crossing each other. This kind of crossing over which takes place. This is now would be like double-stranded DNA, which has been wrapped around itself. Then, to deal with this, the other kind of topoisomerase, which is topoisomerase II. Now, topoisomerase II, as I said, topoisomerase II cuts two strands of DNA. So it will cut this strand of DNA here. Oh, no, sorry, not this yellow one down here, sorry. And then pass this strand through there and religate these two together. Turns out, and this is what we didn't talk about, and what one of the clicker questions was about in Dr. Bartlett's lecture, having to do with how all of these reactions, the topoisomerase reactions, interact with each other. It turns out that twisting and untwisting is topologically identical to taking two strands and passing them through each other. And we may have a chance to talk about that later. But the important thing, as far as we're concerned here, is it doesn't matter if you have a topoisomerase type 1 or topoisomerase type 2 to deal with the problem that you have when you pull the two strands apart. You can either have a type 2 topoisomerase where you pass double strands through each other, or you can have a type 1 topoisomerase where you have this looping structure where just one strand loops around the other one. Yeah?
So how are they oriented? Like, are they looking for certain stress on the DNA strands, or how are they finding the sites that need to be cut?
So the question is, how do you find the sites that need to be cut? And do you recognize a stress situation, what's going on there? So it turns out that we may have noticed in one of the replication fork slides, there's actually a topoisomerase sitting at the replication fork. So it's already ready to go there. But also you will find that these topoisomerases will bind to specific structures. And those structures are going to be because you have overwound or underwound DNA. So basically they will detect the specific DNA structure, but also you have them at the replication fork because you know you're going to need it there. Yeah?
Since I don't see topoisomerase on that bacteria versus eukarya table, right there.
Mm-hmm.
Okay, so topoisomerase has the same name, bacteria versus eukarya.
As far as we're concerned, yes. They're the same topoisomerase in bacteria and eukarya. Just topoisomerase, but you've also got type 1 and type 2 in both kinds of organisms. Yeah?
So what determines type 1 or type 2? Is just kind of whatever the fork's feeling at the time, or could they both do the same thing?
So the question is, which kind of topoisomerase do you have at replication forks? I'm humming and hawing here because I don't want to confuse people too much. Turns out you mostly have topoisomerase II there, but topoisomerase I could do the job just as well.
So it's kind of random, essentially? Or is there...
Okay, there are reasons to have topoisomerase II, and there are particular situations where you have to have a topoisomerase II, and we'll probably end up talking about those after the midterm. But yeah, as far as we're concerned here, for replication forks, where you're pulling apart two strands, either one works. Okay, I want to spend the last 5 minutes or so talking about mismatch repair, and maybe a little bit of initiation, but we'll probably just talk about mismatch repair and leave it at that. So as we talked about last time, you've got the polymerase, bopping this alpha helix back and forth, checking to make sure that the structure of the base pair looks okay. If for some reason that is the wrong base pair which gets added, there's a proofreading activity where you can go back, so 5' to 3' replication, 3' to 5' exonuclease activity, but still certain frequency, again, 1 in about 10 to the 7. You incorporate the wrong nucleotide. And then what happens there? So the process is called mismatch repair because you've just put in the wrong nucleotide. It's a normal nucleotide. It's just it doesn't base pair properly with the nucleotide on the other side. That is detected just like all these other things by a structure, because if you don't have the right nucleotides there, the DNA is going to have the wrong structure. So how do we know about this? Well, partly through studying E. coli, because people found E. coli that put in way too many mutations. And so they figured out that these particular E. coli had mutations in genes that were important for repairing mutations. What do geneticists call things that they've broken? They'll give it the name of whatever it was. So if you have a strain which is making a lot of mutations, you'll call it a mute strain. So a mute, and then the gene that's mutated will be called mute something. So that's what these particular genes are. Here, mute S and mute L. Mute S detects the structure being wrong, and then what mute L does is it's an endonuclease. It will make a cut in the DNA next to this mismatched base. Makes a cut in the DNA. Now you have a piece of DNA which has a primer, a template, and this could be repaired. So this is great, except how do you know, if you're a replicating E. coli or a eukaryote, which of the nucleotides that got put in is the wrong one? Because it's a mismatch. But how do you know whether it's the new strand or the old strand? If it was a mistake that got put in by the replication machinery, it's got to be on the new strand. But how do you know which one's new? You're not measles in a stall and you haven't thrown in light nucleotides at the time. How do you figure that out? Well, it turns out this is actually slightly different between eukaryotes and bacteria. Bacteria methylate their DNA, and methylate it very specifically. And what you have is methylation that happens on both strands of the DNA. Soon as you have helicase pulling the two strands apart, you'll make new DNA, but it takes a while for that DNA to become methylated. And so the way that E. coli knows which strand is the new strand and which strand is the old strand is the strand that's methylated is the old strand. The strand that's not methylated is the new strand. So you know which strand that mute L needs to make its endonuclease in, and that way you end up with an appropriate piece. Eukaryotes basically seem to take advantage of the fact that it's certainly on the lagging strand. You've got lots of individual Okazaki fragments, which are relatively short, and those also will all have nicks in them because the ligase hasn't had a chance to hook up all of your DNA strands yet. How that works on the leading strand is still a very open question. We had a guy a couple of years ago came in the biology department, gave one of the seminars. I'm talking about exactly how that works. But there are a number of proteins that are involved in this process, not just in E. coli, but this protein down here, HNPCC. What's HNPCC stand for? It's hereditary non-polyopsis colon cancer. So it turns out that these mismatch repair proteins, which are not just found in the E. coli that put in a whole bunch of mutations, but also in cancer cells, where people have a predominance of cancer. Again, hereditary, this is something which is inherited. Very high rates of colon cancer. It turns out it's because they have a mutation in one of the genes that's important for making this particular mismatch repair. So we'll stop there. Again, review on Monday and exam on Wednesday.